Okay, so I'm going to go over a couple of things that we talked about in class because I don't think that everything recorded correctly. The first thing we're going to talk about is the syllabus. So if you go to our website, the syllabus is under Course Home, and then there's a tab that says Syllabus, and it should link to this document. So genetics, we have both a face-to-face lab and lecture, and we also have one online student. So we're going to be working with both types of things. I have office hours listed here. Realize that you can always contact me by email. I can always arrange Zoom meetings if you would like a Zoom meeting. And I'm always available to answer questions. So things that you should know about this class. First, we have a required textbook. Please read the text before coming to class. Every chapter has SLOs associated with it. Some of them we'll talk about in class, and some of them you should learn from the book. We have lectures twice a week. We have lab once a week. All of the labs can be done as online versions. Even the things that we do in class, I'll have some online version for. So if for some reason you're going to be gone for a few weeks, please let me know and we'll talk about the online version. Otherwise, I expect that all of you in-class people should be present at lab. Most labs are due the day after the laboratory. A few of them are due a little bit longer. The one that we're doing this week will be due on Tuesday of next week. Okay, here's the learning objectives. You can read those, please. And then here's the schedule. So the schedule basically will go through chapters either 1 through 5 or 1 through 6, and we'll get to the first test. We'll see how we're doing at that point. This is a lot. The first bunch of chapters is all about Mendelian inheritance and other types of inheritance, and it's a lot of problem sets. So for the first exam, there'll be a lot of problem sets. The second exam, there'll be a lot of information about molecular genetics. And then the third exam is a lot more molecular genetics. The fourth is a lot of biotechnology and population genetics, with a little bit of stuff about ethics and other things along the line. Lab is designed to go along with lecture, and the way that this works is if you fail the lecture, you also fail the lab, and vice versa. So please take it seriously. You shouldn't fail either one of them if you come to class, do everything that's required, and then study for the tests. But if you do find yourself being in trouble, please let me know so that let me know early so that we can kind of figure out what's going on. So here's the grades. The labs are worth 25% of your grade. There's a little bit of points for homework, a little bit of points for quizzes. Every week we will have an online quiz that's due on Sunday night. And I made them online so that we don't have to have a lot of papers going through the class. Exams are done in person and they're worth 50% of your grade. And then there's a project, and you can see that the project's due down here in the middle of November. It'll be due in lab. And basically what it is, it's going to be a presentation and a little bit of a write-up having to do with personal genetics, both with the ethical aspects of personal genetics and with the idea of how single nucleotide polymorphisms or small mutations can impact your predisposition to certain diseases. Okay, we have 3 100-point exams and 1 final exam that's cumulative. Honestly, all of the tests are a little bit cumulative because you can't forget what you did on test 1 in order to get to test 2 and 3 and 4. Okay, so that's basically it for our policies. The other things that you should realize, so if you look at our site, I'm going to go back to our course home. All of the information for this site is listed under each unit that's listed under here. So here's transmission genetics. And transmission genetics, basically, chapter 1, the SLOs for chapter 1, the lecture. There will be a class video associated with the lecture. And then there's a link, in this case, to a class forum. There might be links to assignments or other things. All assignments should be uploaded to the assignment box when they're done. which is usually linked to right here. And all quizzes should be done under quizzes and tests. Usually there's a link to them up on this area, but in case there's not, assignments are listed here and tests and quizzes are listed over here. Okay? So having quickly done that, let's look over the slides for this week's class. Okay, I'm going to stop that share. I'm going to go back and look at this one. So I'm going to start this as a slideshow. And just to make sure that it's working, I'm going to go stop share, share it again, because sometimes it misses the fact that it's a slideshow. So here we are with the slideshow, and we're going to start off talking about genetics. So if you look at this front page over here, the thing on the right over here is a karyotype. That's looking at all of the chromosomes in the cell. We will talk about that a little bit later on. To the left here, these are transgenic mice. In other words, they are mice that have a gene inserted into them. In this case, it's a gene that makes the mice glow green. We'll talk about that a little bit later on. And this basically just shows how chromosomes condense. So there's a lot of things, aspects of genetics that we will look at. So we already talked about the syllabus. This is chapter 1. So we talked about this in class, one way that genetics affects your life. We all have some genetic predisposition to certain things. A lot of us have genetic diseases that run through our family. Genetics basically influences everybody's lives. As we get into the future, we are thinking about, we should think about things like genetic engineering, genetic cures for diseases, and also things along the lines of personal genetics, which can also affect the way that we live our lives. So we talked about this on Tuesday. Just in order for us to get to know each other, please submit your information into the forum by August 19th. When I'm recording this, it's actually after August 19th, but if you didn't do it, you can go back and do it. Okay? So why should we study genetics? We are all related to every other life form on Earth. In fact, most of our genes we have in common with the chimpanzee and with other great apes, great apes. So we are very much related to other organisms on Earth. Even the fruit fly have very similar genes to us. We use a lot of these guys as model organisms to study because they're easier to manipulate. We can manipulate their genes, we can make genetic changes, and then we can figure out how those genetic changes affect things like diseases, or we can use them to find treatments for the diseases. So the science of genetics basically allow us to understand how we are similar and how we are different from other organisms on Earth. They also basically, understanding those differences allow us to understand then what particular genes work and how they work in different organisms. We have used the fruit fly, for example, to study diseases, even things like autism and Alzheimer's disease. We've used zebrafish to study human development because they have a very similar development pattern to us. So we can use other organisms by understanding their genetics. We can use other organisms as models to study processes that occur in humans. If we look at the history of genetics, we've been using genetics for a long time. We've been using it to do selective breeding, but nobody really understood what genetics was and how it worked before Gregor Mendel. And Gregor Mendel comes up in the 1860s, though his works was really not that well understood and that well publicized until the 1900s. He's really a contemporary of Darwin and Wallace, who developed the theory of evolution. And Darwin and Wallace's observations on speciation really would have made much more sense if they understood Gregor Mendel's ideas about genetics. But obviously neither one of those was in contact with the other, so they had no idea how those two things came in common. So Gregor Mendel was an Austrian monk, and what he did was he studied pea plants. He started studying mice actually at the beginning, but the mice, they overrun the monastery. Nobody really wanted them. So he moved over to pea plants, and he could study pea plants very easily. What he found, without really understanding how genes worked, he found that there were different forms of particular genes, and that those different forms encoded different traits. We now think of those as different alleles or different forms of the same gene. So for example, we have a gene for eye color. And within that gene for eye color, you could have blue eye color or brown eye color. Okay, that blue eye color and versus that brown eye color are two different alleles of that same eye color gene. So, so if you look at this, here's the timeline down here. So there's Darwin with natural selection in the 1850s, Mendel's in the 1860s. You can see that basically Fleming, a little bit later on, discovered that we had chromosomes. Oops, sorry. Morgan eventually discovers that genes are on chromosomes and genes are linked to chromosomes. But it's not until the 1950s that we understand that DNA is the thing that the genes are on. Once we understand that, and that's really going to be the Hershey-Chase experiment that we'll talk about later on, but once we understand that, there was a race to discover what the structure of DNA was. And that race was won by these two young men, Watson and Crick. I think Watson's still alive. Crick died not all that long ago, actually, maybe 15, 20 years ago. But they were very, very young in the 1950s, and they discovered the structure of DNA. Once they discovered the structure of DNA, it was very easy to see how DNA replicated and how it was transcribed into RNA. Okay, so here's the structure of DNA. We'll talk about this in detail when we get to chapter 10. But what you should remember from previous work is that there are 4 bases, T's and A's and C's and G's. T's always match with A's, C's always match with G's, okay? And then when you replicate this, you have that matchup coming again. These bases are made of, they're nitrogenous bases, and they're attached to a sugar, and that's attached to a phosphate. DNA is a double helix going one direction on one leg and the other direction on the other leg. And we'll talk about that in much more detail when we get to chapter 10. The other thing that you should understand that runs through this entire thing is the central dogma of molecular biology. And the central dogma says that all cellular organisms, not including viruses, but everything that has a cell, the genetic material is DNA, and that genetic material gets transcribed into RNA and translated into proteins. The DNA replicates itself, so replication is something that DNA does. It gets transcribed into RNA using RNA polymerase and translated into proteins using the ribosomes. Everything on Earth does it this way. Some viruses will use RNA as their genetic material, and a few viruses can do reverse transcription where they make DNA from RNA. But everything that's a cellular organism, not including viruses, will use this central dogma. Okay. Other things that you should understand, when we study an organism now, we think about the genome, or all the genes that are in the organism, and the proteome. And the proteome basically are all the proteins that are expressed in the organism. So the genome are all the genes. The proteins basically are the genes that are expressed as they get translated into proteins. Genomics studies all the genes, and proteomics studies all the proteins. Those are really recently developed studies based off of us knowing all the genes that are in certain organisms, including humans. So genomics and proteomics are recent studies, but they're telling us a lot about how things change, particularly how things change with disease states, or how things change with different gene expression. As we do this, we can use genomics and proteomics basically as a way to develop a better understanding of the entire organism. Mutations are also important. Mutations are changes to the DNA. Many of them get repaired. So most of them occur during replication, with a few of them occurring because of chemical or physical mutagens or things that mutate DNA. In the human system, we have large genomes, but much of those genomes do not affect, most of those genomes do not code for proteins. So mutations can occur in regions that code for proteins, or they can occur in regulatory regions, but a lot of them actually occur in areas of the genome that don't code for anything as far as we can tell. Some of those mutations, if they're small, we call them single nucleotide polymorphisms. Those are differences in DNA sequences between individuals that occur reasonably frequently. Many of them are not associated with any bad outcomes, but some of them are associated with disease outcomes or different traits between individuals. So single nucleotide polymorphism, SNP is the other name for it, SNPs. Those differences are will end up being fairly important, and they allow us to characterize different individuals. When we talk about genetic genotyping, which we'll talk about later on, one of the things that people look at are those single nucleotide polymorphisms. A molecular clock basically looks at the differences between two different species by postulating that the more mutations, so the more differences that you see between species 1 and species 2, the higher the divergence between those species, and the longer the time since those species had a common ancestor. And you can basically see that mapped out in a phylogenetic tree. Okay? So genetics and evolution, way back in the 1860s, talked about the accumulation of differences over a long period of time. And that was first elucidated by Charles Darwin and Alfred Wallace, working independently. They didn't really understand genetics, so they didn't really understand how those mutations worked, but they realized that over time, species diverged from one another. Now we can actually map how those species diverge, and you can find an evolutionary map, which can be seen on a phylogenetic tree. That way you can see and quantify the evolutionary changes and see the differences in population over time. This site right here shows you how those divergences work in real time for the new coronavirus. So in organisms that diverge quickly, like viruses, you can see that happening over time, even over a period of months. With most organisms, you're seeing that over a period of millions of years. So we're going to talk about 3 different types of genetics when we go through this course. The first one that we're going to talk about is transmission genetics. And transmission genetics basically asks how genes are inherited. It talks about dominance versus recessiveness. It looks at pedigrees of families. So here's a pedigree of a family showing the inheritance of a genetic trait. And it looks at how people are related. This is a Punnett square down here showing the alleles coming from mom and coming from dad. With transmission genetics, you can do genetic genealogy, which is where you get your own genome sequences done. What they're really looking at in this case is going to be single nucleotide polymorphisms. And you can see here, this is my DNA. All these purple areas are areas that I share with a second cousin. And when I contacted the second cousin and we talked about it, we realized that we shared a set of great-grandparents. So this genetic genealogy was correct in terms of it predicting that that person was the second cousin. She really was the second cousin. She was born in the area where my mother's relatives were born. And these are the areas of the genome that we share. Now this is only showing one of the 2 sets of homologous chromosomes. So it's really not showing you both sets, but she is really on my mother's side. So this would be my maternal set of chromosomes. You can use transmission genetics to predict relationships. I've used it before to help people find their birth parents. I had a gentleman who was a client of mine who um, was 57 years old, and his wife lovingly gave him a Ancestry.com DNA kit, and through that DNA kit, he discovered that none of his known relatives showed up on that site as being his relatives, and pretty quickly he discovered that he had been adopted. He never knew he was adopted before. So these can bring out surprises for people, finding out that you know they had problems, or that they were adopted, or that they have predisposition to genetic diseases. They can find out all sorts of things. People find out that their fathers were not their fathers, or at least not their birth parents. So genetic genealogy can be beneficial, but it can also be problematic. You can also use genetic genealogy to find the perpetrators of crime, or to identify bodies. So this is often used by law enforcement as well. And that all comes under transmission genetics. Molecular genetics looks at the DNA itself. And molecular genetics basically looks at DNA and protein, looks at form and function. So in other words, structure, how the protein is structured, and how the structure relates to function. Molecular genetics also allows us to manipulate genes in a lab, and allows us to look at how genetic defects affect proteins and cells and the physiology of organisms. A good example of molecular genetics at work is Pompe disease. So Pompe disease is a very devastating disease. Basically, it's a buildup of glycogen inside the lysosomes and muscles. And when that builds up as you're using the muscles, it eventually destroys the muscles. And children with this disease generally live into their teenage years. By that point, they're in a wheelchair, and after a while, their muscles completely stop working, and they can no longer sustain life. So people with this disease had a fairly progressively bad disease, and then they would die in their teenage years. It turned out when this disease was studied by molecular geneticists that there's a mutation in an enzyme that makes this particular, a mutation in a gene that makes this particular enzyme. This is the enzyme that usually breaks down glycogen. So glycogen would build up in the lysosomes of muscles, and basically what happened then is that the muscles would get destroyed. Using that information, what the molecular geneticists did is they basically took the gene for this enzyme, the normal gene for this enzyme, put it in Chinese hamster ovary cells, and because the genetic code is the same between those Chinese hamster ovary cells and human cells, those hamster ovary cells made that particular protein, and that protein could be isolated and purified from those cells. It then could be injected, infused into people with Pompe disease, and that's actually a cure for Pompe disease. It's a cure for Pompe disease that can allow those children to survive and live more or less normal lives. So every month or so they have to get this new injection, but once they do that, they can live pretty much normal lives. So that's an example of the power of how molecular genetics can cure a disease and save a life. Molecular genetics also encompasses model organisms, and since pretty much almost everything on Earth uses the same genetic code, and pretty much everything on Earth has some similar enzymes, we can use model organisms from bacteria to yeast to mice, zebrafish, plants, monkeys. We've got fruit flies here. We got zebrafish. C. elegans is a worm that's been used a lot. This is a mouse. Arabidopsis is a plant. All of those have been used as model organisms. They have very similar functions to us, so we can use them to study processes. So for example, yeast have been used to study the cell cycle because their cell cycle is very similar to ours. Mice you can use to basically mimic a genetic disease. And then you can watch the natural progression of that genetic disease. You can test drugs that eventually you want to use in humans. You can test them on mice. You can study the gene's function by knocking out a particular gene, or you can make mutations that mimic a genetic disease so that you can study the genetic disease. Okay. Our third thing that we'll talk about towards the end is population genetics. And population genetics basically looks at how allele frequencies change over time in different populations. This can basically see how differences in environmental factors can impact the expression or the allele frequencies of a particular gene. One example of this is looking at the allele frequencies and the prevalence of particular genetic disease. So the example that your book gives in the first chapter is albinism. Albinism is a disease that is spread, is a recessive disease, so you need to have 2 alleles of that particular gene in order to be affected. But we know that the prevalence of this gene is different in different populations. The Hopi tribes in the 1800s considered a person with albinism to be very lucky. People with albinism suffer from skin problems, and they suffer from an inability to see very well. But because they were considered lucky, the Hopi tribe down in New Mexico would make sure that albino individuals were protected from the sunlight, and that they were given special tests that kept them out of the sunlight. And that allowed them to live normal lives. They would breed, they would have children, and over time, the incidence of albinism came to be about 1 in 200 because people really wanted children with albinism. On the other hand, in Tanzania, the incidence of albinism is much lower, and that's because, and partially that's because children with albinism are considered to be very unlucky. Sometimes they're abandoned, sometimes they are killed off, and sometimes their body parts, they were killed for their body parts to be used in witchcraft. So the incidence of albinism over time in Tanzania really went down as people with albinism were not considered to be were not protected and were not considered to be a good outcome for children. The Human Genome Project is a thing that we'll talk about. This is basically a publicly founded, large project to sequence the entire human genome. I realize that's a lot of nucleotide pairs, but we found that there's actually very few genes in the human genome, about 25,000 or so. We always find a few more, but there's not going to be huge numbers more than that. But what that has allowed us to do is take genomics and apply it to the human population. This is basically the science behind the use of personal genomics, 23andMe and other places. It has a lot of ethical issues because you have to think about if we understand how personal genomics affects your life, we could use that to discriminate against people. Also, it's, it might be used against people that privacy. They might not have the privacy of the genetic information. If your genes are passed down to your children, knowing your own genomics may cause problems for your children. So there's lots of ethical issues that we'll talk about here. But the Human Genome Project really was a good thing in terms of moving us forward to understand how our genes and our proteins are related to one another and how the genome basically encodes the proteins that it does. Genetic applications, of course, are found all over the place. And we think about this in terms of biotechnology. We've used genetics in agriculture from the beginning. All the selective breeding that was done in the old days was application of genetics, even though we didn't understand that was what it was. Medicine is using genetics a lot. And of course, in society, we see it a lot as well. So in agriculture, in the early days, there was a lot of selective breeding, leading to us basically developing things like corn and other organisms like wheat, which are much different than their wild cousins. Now we use selective breeding, but we also use genetic manipulation to make genetically modified plants. We use molecular genetics to understand how plants change over time and to basically help us to select the best plants for feeding the nation and feeding the world. Medicine, of course, is the study of diseases. We can use genetics to study genetic diseases. We can also use it to study diseases like cancer that end up, even though they're not inherited, they end up with a lot of genetic changes. You can use it for genetic counseling. So if you know that you have a genetic disease running in your family, you can decide what you want to do as you have children. And also we use it in individual lives. So one place that it's used is some people have a gene that's an allele of BRCA. BRCA is a gene that is found in breast cancer. BRCA is a gene that everybody has, right? But some people have polymorphisms in one allele of their BRCA gene or two alleles of the BRCA gene. And if you have these polymorphisms, it predisposes you to breast cancer, okay? If you have those polymorphisms, what a lot of women elect to do is to have breast cancer screenings early. Some women may elect to have mastectomies because that will eliminate their ability to have breast cancer. If you know that you have this gene, it allows you to make particular decisions that you would not be able to make otherwise. Medicine, basically, you can use genetics now to treat disease. One way to do that is to have use cells, either mouse cells or cultured cells or bacterial cells to grow drugs that are then used to treat disease. So one example of that is insulin that's used for diabetics is grown in bacteria. It's been grown in bacteria now for any number of years. So when you buy insulin, inevitably and always, usually it is grown in bacteria. Other ways that we can use this is basically using molecular genetics to discover what the protein target of a drug should be. And if we can do that, basically what we can do is we can discover how to develop drugs against that protein target. The Pompe example was a good example of that. We're moving towards human gene therapy. Some of the first gene therapy trials have been in the past couple of years. And basically those are used to insert a healthy gene into somebody that's diseased, that has a genetic disease, or to remove a bad gene from somebody that has a genetic disease where the bad gene is dominant and causes problems. Pharmacogenomics is another example of medicine. Pharmacogenomics basically is the study of drugs and how people's genetic codes interact with those drugs. What basically it is it tells us how different drugs are metabolized by different people. So some people will metabolize them very well, and some people won't metabolize them very well at all. If we understand that, we can know which drug to use to help treat different human diseases. Okay. In society, of course, we see this in agriculture, we see medicine, all of those have societal impacts. And in addition to that, there's a lot of economic impacts of the biotechnology industry. There's lots of economic impacts of pharmaceutical industry. Legal analysis is important, so finding paternity tests, testing inheritance claim, looking for the perpetrators of crime, IDing bodies, all of those basically are going to use genetics. And of course, at this point, we come down also to moral issues. Moral issues go back to the time of eugenics, when we first understood how genetics worked. People wanted to use it to determine who should breed with whom in human genetics. So there's lots of moral issues in society that spring from genetics. Okay, so organization of class. The first week we'll talk about cell cycle and mitosis and meiosis. After that, we go into a section on transmission genetics. Then we talk through molecular genetics. After that, we talk about biotechnology and genetics and society. And then we end up at the end of the course talking about population genetics. Unfortunately, we don't really have as much time on that subject, but it is a very good subject. So we'll go through all of these and hopefully finish up on a high note in this class. As always, please talk to me if you have problems along the way. My aim is to be really flexible this semester. I know that we're going to have people out because of the pandemic, and I know that there may be some problems along the way. My aim is to try to make sure that we all get out of this alive and that we all get out of this with the best possible learning experience possible. So please keep in contact with me no matter what. I'm going to stop this and move on to the next section. So I will see you in class later on, or I will be in contact with you later on.